Plasmolysis. This is the process by which plant cells lose water, shrink, and become flaccid, with the cell membrane detaching from the cell wall. If a plant cell is placed in a hypertonic solution, that is the one that has a higher solute concentration, more than the cytoplasm and the subvacuole concentration, then water molecules will move from the cell by osmosis into the hypertonic solution. And as this goes on, the cytoplasm together with the subvacuole will reduce in volume. If the loss of water continues, the cytoplasm and the subvacuole will continue reducing in volume to the point that the cell membrane becomes detached from the cell wall. Now, such a cell is said to be flaccid, one that has lost its turgidity. Remember, turgidity is achieved when the turgor pressure equals wall pressure. But as a result of plasmolysis, there is loss of turgidity, and with that loss of turgidity, the cell becomes flaccid. So you talk about a flaccid cell, one that has lost its turgidity. Now, as the shrinkage of the cell continues and the plasma membrane detaches from the cell wall, the cell becomes flaccid, or it's also said to be plasmolyzed. The cell is said to be plasmolyzed when it loses its turgidity. Now, when the water loss continues, you have more and more water being lost by osmosis. Total plasmolysis is achieved, and this occurs when the cytoplasm shrinks so much such that it just forms a mass, a spherical mass at the center of the cell, and the space between the cell wall and the cell membrane will be occupied by the solution, that is the hypertonic solution in which the cell is immersed. So all of this will consist of the hypertonic solution, because remember, the cell wall is freely permeable. So the molecules in the hypertonic solution will freely pass across the cell wall and get into the space between the cell membrane and the cell wall.